So last time we started our exploration of everything that dogs can tell us, and we're going to continue that today. Last Wednesday we were focused on the question of how DNA sequences can be used to tell the history of a group of organisms. And one of the things that we found last time was that even though we could figure out, say, that gray wolves are the ancestors of modern dogs, just having a little bit of sequence from the mitochondrial DNA of a bunch of dogs was not sufficient to actually tell those dog breeds apart. You couldn't tell a poodle from a German shepherd, and in fact you couldn't tell a dog from a wolf in some cases. And so what we're going to talk about today is how people figured out how to do that better, and actually now you can make a DNA test basically with very, very high accuracy that will tell you what breed your dog is, which may or may not be a useful practical application of DNA technology, but that's for you guys to decide. But what's important to us is that it actually tells us a lesson about how that kind of information can be used, how DNA sequences can be used to figure out the relationships even between very, very, very closely related individuals. And the other thing we're going to start talking about today, which is a really important theme in the course, is not just anonymous DNA sequences. So like if you remember from when we were talking about the universal tree of life and eukaryotes and bacteria and archaea, the piece of DNA that was used to figure out that tree was a bit of the ribosome, okay, DNA that encodes ribosomal RNA. And that was very useful for that purpose because all organisms have ribosomes, all organisms have that particular ribosomal RNA, and that's why it had to be used. But that gene is pretty much the same in all organisms. That's why it was useful. And so it doesn't tell us about why organisms are different from one another in how they appear and how they act and so on. And so what we're going to start talking about today is how DNA sequences can be associated with traits, how knowing that a particular gene is different between two individuals tells you something about why those two individuals look different. Okay? And dogs, again, present a really good example or set of examples about that. Okay? So those are our topics for today. And to summarize them in questions, what we're going to discuss is, can a DNA test tell what breed a dog is? The answer is yes, and you'll see how that works. How do differences in genes lead to differences in complex traits? This is the sort of central question in genetics and genomics. And how is dog genomics related to human disease? I think I mentioned this last time, one of the reasons people are studying dogs so intensely now is not just that they're cute and lovable, but that they can tell us something about our own species, and in many interesting ways. And it turns out that one of the ways dogs are very similar to our own species is that they suffer from a large, large number of genetic diseases. And so that's where I want to start today. And so there's this kind of upsetting website that exists that sort of catalogs. It's not upsetting because it exists. It's sort of upsetting to read how terrible some dogs' lives are. It catalogs all of the genetic diseases associated with particular breeds, okay, and it just goes alphabetically. So the first breed is the Affenpinscher, and you can see that there are certain predispositions within this breed for a large number of diseases, okay? For example, cleft palate, right, which is sort of malformation of the upper lip, lots of other things, and so you can sort of go down the list and see some really interesting cases. So I just want to switch to the actual website. So here it is. I just want to go to you can see this is really sort of a litany of really horrible things that dogs have to suffer through. Does anyone have a good explanation for why dogs are so plagued by genetic diseases? If I did this for wolves, how many genetic diseases would we be able to count on? Yeah, so inbreeding has an enormous amount to do with this. Because many diseases are recessive, that is, it takes two bad copies of a gene to actually show the disease phenotype, right? And so one way to get homozygous recessive individuals, that is to get 2 copies of a bad gene, is that if you have those 2 copies of that bad gene because it traveled down 2 sides of the same family, okay, some ancestor had the bad disease gene, and then in a simple case, for example, it passed down to a brother and sister. If those brother and sister have children, then it's much more likely that the offspring will then be homozygous for that than if the brother and sister had mated randomly in the population. Okay, and so because the way breeds are created is by serious inbreeding, mating brothers and sisters over and over again, you get these situations, unavoidable situations, where bad copies of genes come together and you see these manifestations as diseases. So some of the diseases you see are exactly analogous to human diseases. For example, Dalmatians, where are they? Are prone to glaucoma, okay, eye disease, right? Muscular dystrophy, a lot of other things, right? Doberman pinschers are really interesting. They're prone to narcolepsy, okay? So Doberman pinschers tend to just sort of fall down asleep, right? And these are human diseases as well. And the hope is that by understanding the causes of the diseases in dogs, then we might have some way of understanding the causes of the diseases in humans and doing something about it. So that's even sort of more indication, because these diseases are sort of specific to particular breeds. Dobermans are prone to narcolepsy. Not every dog breed is prone to narcolepsy. In fact, I think it's only Dobermans that are prone to narcolepsy. So that tells us that there should be genetic differences between these breeds that are diagnostic. We should be able to find pieces of DNA that say, this is a Doberman, this is a poodle, this is a German Shepherd, okay? And as I said at the beginning, up till now, we haven't really seen how that works, because if we just take mitochondrial DNA, for example, and this is just a slide from last lecture, you can see that a German Shepherd might be sort of all over this tree, right? One German Shepherd down here with exactly the same sequence as a wolf, one German Shepherd up here, one German Shepherd up here. It's not like the German Shepherd sequences are all similar to each other. So that means that this piece of mitochondrial DNA is not sufficient to use as a DNA test to tell you what dog you're looking at, okay? And so to summarize why the previous studies like that one couldn't tell breeds apart, there are basically 3 main reasons that were thought to be at work, okay? One reason is maybe it wasn't enough DNA sequence. Maybe you need a lot of DNA sequence so that these rare differences show up that are the diagnostic ones that allow you to tell the breeds apart. Okay, so maybe just by sequencing a lot more DNA per individual, that would solve that problem. Another problem, as you saw, is that in some cases, the wolf sequence and the dog sequence were identical, okay? And what that essentially means is that the mutation rate is not fast enough for a bunch of changes to have accumulated between those sequences. So maybe if mutations were faster, then we'd be much better able to tell breeds apart easily, because there would be more changes to look at. And again, these diagnostic changes would be there. Okay, so we can't go back and to the origin of dogs and say, okay, the mutation rate should have been faster, right? That's not something we can do. But what we can look for are particular DNA sequences that are prone to mutate faster, and maybe those could be used for this kind of analysis of really close relatives. And then finally, a really important thing to consider, which we talked about a little bit last time, is that maybe this branching tree is not the perfect reflection of the history of these organisms. So I talked last time about how if you're only tracking mitochondrial DNA, you're really only tracking the mother of the mother of the mother of the mother of the mother back in time. And if you're only tracking the Y chromosome, it's only the father's 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 father. And so that doesn't capture the entire history of that organism. And in fact, the farther back you go in time, the worse it is at capturing that whole history. Okay? And then the other problem you have with members of the same species is that they might be crossbreeding with each other. Okay? And so you might have a pure breed of poodle and a pure breed of German Shepherd, but all it takes is one sort of promiscuous poodle to cause the problem where you can't identify which breed you were looking at anymore because then the DNA sequences mix. Okay? And so when you have crossbreeding, a tree isn't really the right representation because a tree implies that these sort of changes are happening and they're kept separate. Crossbreeding puts them back together. Okay? And so all three of these problems can be addressed, and they were addressed in a recent study. Okay? And what I want to go through is sort of how they did it. Okay? So the way they dealt with this problem of having not enough sequence is they got more sequence. Okay? That, in a way, is the most straightforward thing to fix. Okay? There's a whole genome at your disposal, so why not just go gather more sequence information and not just limit yourself to mitochondrial DNA, because then you'll be getting other chromosomes, you'll be getting sort of more of the history reflected in the sequence you're actually looking at. This problem of mutation being too slow is something we're going to talk quite a bit about today in terms of how certain DNA sequences are just prone to mutations and what problems and opportunities that causes. And so there are particular sequences called microsatellites, and we'll talk about what this means in a second, that are known to be fast mutating. That is, if you have just a random stretch of sequence A's, T's, C's, and G's, you know, mutations happen very rarely in it. But there are certain sequences in your genome, in every sort of organism's genome, like dogs and so on, that are prone to change fast. In fact, they change in the course of a single generation. Every generation, that sequence mutates. Okay, and we'll talk about examples of these, because not only are these sequences really important for reconstructing recent history, but they turn out to also be associated with observable differences between individuals, both diseases and other traits that we might be interested in understanding. Okay, so we're going to talk about these kind of sequences in a second. And then the other thing that this new study did is they said, okay, trees maybe aren't going to cut it in terms of telling breeds apart because of that German Shepherd issue. They just sort of fall out in different places of the tree. And so we need some newer, better approach to deal with that. And they use something called machine learning, which I'll talk about in a second and explain what that means. Okay? But basically what it does is it says, we're not going to try and fit a tree. We're going to try and use some special statistical computational approach to deal with this problem. Okay? So I want to tell you a little bit more about microsatellites. They're actually very simple. So microsatellites are basically when you have a short sequence that's repeated over and over again. And you've seen that DNA sequences, sort of your genome, for example, are plagued with repeats. Okay? And so when you have especially small repeats, like this is a 3-nucleotide repeat, CAG, 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 that's called a microsatellite. And it turns out that the enzyme that copies DNA, DNA polymerase, which you all know about, tends to have trouble copying these things. One way to think about it is if I asked you to count to 3 10 times, okay? You'd go 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. Wait, was I on 4 or 5? Okay? It's harder to count to 3 10 times than it is to count to 30. Okay, because you lose track of which repetition you're on. Okay, DNA polymerase, in a sense, has the same problem. When its job is to copy triplets that are repeated, it has a problem. And you'll see in the next slide what the problem actually is physically. But because these sequences are common in genomes, and because the DNA polymerase has a problem with these sequences, there are certain reliable sets of sequences in the genome that you can count on to mutate very frequently. And you can use those to, for example, reconstruct the recent history of a group of organisms. Okay? So why does DNA polymerase have a problem with sequences like this? It actually, in a sense, gets confused. Okay? And so this is what it looks like. Imagine that you have one strand of DNA, which is written in the 3' to 5' direction, GTC, GTC, GTC. So the DNA polymerase is going to come along and try and make a copy of this in the 5' to 3' direction. Okay? So basically, it's going to add CAG, 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 and try and keep going. Okay? That's what normally should happen and will happen most of the time. Okay? But occasionally, as I said, it gets confused about what repeated that it is on. And the reason is that this pairing of CAG to GTC, CAG to GTC, is going to be followed, but you can see that this CAG could pair with this GTC. Okay, these are just two complementary strands of DNA. And so what can occasionally happen is that this newly replicated strand, for example, can loop out, and the sequence will just keep going because this CAG, which was copied off of here, now sort of backs up and goes back to here. Okay? These are sort of two physical objects, and they have this matching every 3 bases. And so once this looping happens, it's stable again because CAG is paired to GTC, CAG is paired to GTC, and it can just keep going. But because the loop happens, then when this copy of DNA is finished, instead of just having repeat 1, repeat 2, repeat 3, repeat 4, repeat 5, repeat 6, what you have is 2 copies of repeat 2. So it goes 1, 2, 2, 3, 4, 5, 6. And so you started out with 6 repeats, and now you have 7 repeats. Okay? Because the DNA polymerase sort of backed up 1 repeat and copied it twice. Does that make sense? Okay? The same thing can happen if the template strand, the one that's being copied, loops out. Okay? So in this example, what you have is 3 repeats and then a loop and then 2 more repeats. And if the DNA polymerase just sort of goes straight across here and doesn't copy this one because it's sort of doing just fine here, everything is paired, then the result there is that you shrink. So you get 5 repeats instead of 6. You're missing now this 4th repeat. Okay? So in general, what happens when DNA polymerase reaches a repeated sequence like this is either it stays the same because replication actually happens properly and it just goes straight across, or occasionally it slips backwards and then you get more repeats than you should, and occasionally it slips forward and you get fewer repeats than you should. Okay, so the number of repeats can change basically each time the DNA is copied. All right, so that's the fast mutating sites. Now I want to talk a little bit about machine learning, which is the other element of trying to figure out a DNA test for breeds. Okay, we need some better way than a tree to figure out how these organisms are related to each other. Okay, and so machine learning sounds like a sort of complicated, fancy thing, and in a way it is, but it's actually sort of something that should be familiar to you already because we live in the internet age. Okay, and so the sort of components of machine learning are, the first thing you do is you gather lots of data on individuals of known identity. Okay, so in this example, what we do is we get DNA sequence data for a lot of dogs where we know their breed, and the breeding records are really good, and we know it's purebred, and so on. So you gather lots of data. And then you do something a little bit mysterious. You use complicated statistics to find patterns in these data. Okay? And what you're looking for is patterns in the data that are associated with these known identities. Okay, and I'll give you a familiar example of this in a second. And then once you've figured out the patterns, then you don't need the identity anymore. If someone gives you a new individual, you can look at which pattern it fits the best, and then you assign it that identity. Okay? And so here's a familiar example, I hope, to you of machine learning. Okay? It's how a website like Amazon.com recommends books or movies to you. Okay? So here's the way it works. They have lots of data. Basically, they have data on every single purchase that's ever been made on Amazon.com. Okay? And so we can think of that data as sort of dogs as customers and their DNA sequences as all of the purchases they've ever made. Okay? And so the idea is from those data for each user, you can get a pattern, what their tastes are, what they've tended to buy in the past. Okay? And then you look for people who have similar tastes, right? Similar patterns. And let's say there's a book on someone else's list where that person matches me really well, but I haven't bought that book yet, but they have. Then it'll make that recommendation to me. It'll say, you know, you will probably like, this book, Into the Cool, Energy, Flow, Thermodynamics, and Life, okay? You will probably like the Smithsonian Intimate Guide to Human Origins, okay? And these things tend to be fairly accurate, and the more data they have, the more accurate they are, right? Like I cried like a baby during Brokeback Mountain, okay? So the point is, when you have that much data and you have fancy computer algorithms, you can find patterns really easily, okay? And then you can make recommendations in this case, or in the case of dog breeds, you can sort of make assignments. You are very likely to be a poodle. You are very likely to be a German Shepherd, okay? Does that make sense? All right? And so when you do that, it's amazingly accurate for dogs, okay? So I hope what you can see here in this very complicated figure from a real paper is the dog breed names, okay? So like American Cocker Spaniel, Old English Sheepdog, German Shepherd, Irish Terrier, okay? And they're put in order of sort of how closely related they are as this algorithm determined. And then what's shown here is each breed is in a different color. And the 0 to 100% scale is, once they figured out the algorithm, then they tested it and they asked, well, let's take another set of dogs where we know what breed they are, get their DNA and see if the breed is figured out by this algorithm, okay? So you develop the algorithm and then you test the algorithm. So for example, for the Irish Wolfhound, they gather a bunch of new Irish Wolfhound DNA sequences and they ask, with just that sequence, do we say this is an Irish Wolfhound or do we say it's something else, okay? And the color for the Irish Wolfhound is this brownish color, okay? And what you can see is over 99% of the Irish Wolfhound DNAs that were given, the algorithm figured out it was from an Irish Wolfhound, okay? And towards the ends here, you see these sort of speckles of color. Those are all the misassignments, okay? So occasionally it gets it wrong. And you can see for like the Belgian Sheepdog, there's a few errors made, but only a couple of percent. For the most part, every Belgian sheepdog DNA that's obtained, the computer spits out this was a Belgian sheepdog.
How is it determined what the dog breed is before the DNA is sequenced?
Right. So the nice thing about dogs, one of the things that makes them very useful for testing algorithms like this, is that breeding records are kept meticulously. Okay, if you go to like a kennel show and you bring your French bulldog, they won't let you in unless you have the records showing, what its parentage is, right? What its pedigree is. Okay, and so the breeds are very well known. You can get purebred dogs where the documentation that that's a purebred dog is just fantastic. Okay, so there's no question about it that dog is a bull terrier or is a boxer or whatever. Okay, and that's what allows you to do this. It allows you to have this real test of whether the DNA algorithm is working. Because you're right, if you weren't sure about what breed it was in the first place, then it's not really a test of whether your algorithm is working. Okay, and so in most cases, you can see that these color blocks are pretty solid almost all of the way up. Okay, and what that means is for every particular breed, it's doing an amazing job, like 99% job of classifying just based on that DNA. So something like this, I think fairly could be marketed as a real DNA test of breeds, right? If I give you the DNA of my dog, this algorithm will tell me if it really is a golden retriever or something, okay? And for these kennel clubs, this is something they're very interested in because, they have the pedigree data, but this is sort of the modern incarnation of it. If you can just do a DNA test and tell whether the dog is legit or not. The other thing that's really interesting is some of the breeds that were used are really closely related to each other, okay? So like the Alaskan Malamute and the Siberian Husky are very, very, very similar dogs, but they're classified perfectly. 100% of the time, if it's Alaskan Malamute DNA, then the computer says it's an Alaskan Malamute, and 100% of the time, if it's Siberian Husky DNA, it says it's Siberian Husky. The same for these two, Collie and Shetland Sheepdog, Greyhound and Whippet, Burmese Mountain Dog and Greater Swiss Mountain Dog. Okay, these are Bernese, not Burmese. So these are really similar dogs, but the computer and the DNA analysis can still tell them apart. Here's an example where it's 50-50. So Belgian sheepdog is in light green, and Belgian Tervuren is in dark green. And what you can see is that half the time a DNA sequence will say it's a Tervuren, and half the time it'll say it's a sheepdog. What this means basically is that those probably are not real, actually different breeds. Okay? It's somebody's choice in the very recent past to start calling them different breeds. The other thing that we can see from this analysis is that there's a division between what are called ancient breeds, okay? And the reason they can be called ancient is that their DNA profiles are most similar to the gray wolf, okay? And then there are what are called modern breeds, which derived from the ancient breeds that were originally domesticated, but now we have this amazing diversity of breeds that have been developed over the years. The ancient breeds, what you can see here is a lot of Chinese dogs, for example, Chow Chow, Shar Pei, Shih Tzu, okay, Pekingese. That's because, as you know from last time, the origin of domesticated dogs was in East Asia. Okay, so these are representatives of these really ancient breeds that have been around for a really long time. The rest have been sort of more modern creations. Now, the thing is, a couple of controversies emerged when people started looking a little bit more carefully at these data. For example, one of the breeds over here is the Norwegian elk hound, which looks like that, okay, on the modern side of things. If you go to Wikipedia, which, as you know, is the most reliable source of information out there, and you read about the Norwegian elk hound, this is what it says. It says it's a very ancient breed, having been developed over 6,000 years ago to help early Norsemen hunt big game such as moose and bear. Remains of dogs remarkably similar to the modern elk hound have been found in Viking grave sites such as the cave in Jæren, Norway, where they were dated back as far as 4,000 to 5,000 BC. Archaeological digs suggest this breed existed and was domesticated in the Stone Age. False. Okay, and what do I mean by false? What I mean is that the DNA says modern, right? The DNA says it's not closely related to wolves like these East Asian dogs are. And so what this probably is a modern recreation of the ancient breed, okay? Sometime in the much more recent past, people selected for the same characters as the Norwegian elk hound of fabled lore had, okay, and got it, right? And so now we have this breed that's actually a modern breed that resembles the ancient breed. And a better example of this is actually the pharaoh hound, okay? Egyptian dog, right? So this is an ancient pharaoh hound mummy, right? This is a carving of a pharaoh hound, right, from ancient Egyptian sites, okay? This is a modern pharaoh hound. And you can see how remarkably similar this modern dog is to what existed in Egypt thousands of years ago, okay? You can see the curve of the tail, curve of the tail, curve of the tail. You can see the pointy ears. You can see the really broad chest and narrow waist, okay? This is, by all accounts, you know, this dog, right? The problem is it's not, right? The problem is the DNA says this is a modern breed. It's not an ancient breed. So again, one of the explanations that's fairly likely is that in more modern times, people recreated this breed, okay? Just by starting with a dog that was similar enough to this and then selecting for the traits that are characteristic of this until they get it. They get the curled tail and the pointy ears and the broad chest and the narrow waist, okay? So this sort of leads us into the question of, well, how do genes control these traits? You know, I just told you that the likely thing that happened with those dogs is that you do selection again and you get the traits you want. And so that means that certain genes have to mutate so that those traits appear and someone could select them, right? So the question we want to now talk about, and as I said, is a major, 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 major question in biology, is how do genes determine how organisms look and behave, right? And so again, dogs provide lots of examples that we can go try and figure out because there are so many differences between the dog breeds. that have arisen in a very short amount of time. And so we have some hope of figuring out the mutations that actually occurred that, say, make a pharaoh hound look like a pharaoh hound. Okay. In scientific lingo, what we're trying to connect is the genotype, that is the DNA, the content of the genetic information, with the phenotype, the appearance and behavior of the organism. Okay? So somehow we have to get a connection between genotype and phenotype, right? And we can do that in dogs. We can ask what genes are responsible for the observed differences among breeds, and I'm going to show you a bunch of examples today of ones that have been worked out just over the last couple of years. And in general, how do genes control complex traits like the shape and size of an organism? Okay? And as is typical in biology, to understand these very complicated questions, we start with sort of simple examples. Okay? And so the simple example I'm going to start with is this one. So these are 2 glasses of beer. Okay? This one is clear. This is like sort of an American lager. And this one is cloudy. It's like a wheat beer. Okay? And does anyone know why this one is cloudy and this one is clear? Unfiltered, but what's actually filtered? Yeast. Okay, so this one, you know, when you're drinking a glass of beer like this, and if you're under 21, I assume you're not. If you're drinking a glass of beer like this, you're basically drinking a suspension of yeast. Okay, that's why it's cloudy like this. And if you're drinking a glass of beer like this, that yeast has been removed out. And actually, there are 2 ways the yeast can be removed. One is that it's filtered out, and the other is that it naturally falls out. Okay? And so the yeasts that are used to make these 2 different types of beer are actually different strains of yeast. Okay? And the ones that are used to make wheat beers have this phenotype called not clumpy. And the ones that are used to make clear beers have this phenotype called clumpy. And you can see that there's the same number of yeast cells here and here, except in these test tubes, the yeast is on the bottom. Okay, that's the yeast right there. And the reason it's on the bottom is because the yeast strain that's used to make the clear beer contains cells that like to stick to each other. Okay? Those cells stick to each other, more cells stick to those, more cells stick to those, until you get this big mass of cells, and the density of that mass is heavy enough so that it sinks to the bottom of the tube. Okay? With this beer, you can see a little bit of gravity having an effect and bringing down some of the yeast cells. But because they're not attached to one another, each one is floating free, their density is approximately the same as the liquid, and so they stay in suspension. Okay? So we actually know the molecular mechanism of this clumpiness and not clumpiness. And the way it works is that on yeast cells, so this is a picture of a yeast cell with its cell wall. Somewhere in here would be the nucleus, right? They have a particular sugar called mannose. Okay? It's typical of yeast cells. There's these mannose sugars that are sort of studded in the yeast cell wall. Okay? And there's a gene that encodes a protein that has a pocket in it that exactly fits that sugar. Okay, it's sort of like a lock and key kind of thing, where this protein can line up exactly with the sugar, and they're sticky to each other. Okay, it's the type of protein that works in our immune system to recognize invading pathogens, for example. It's a protein that sits in the cell membrane and faces outside and is ready to receive whatever is there. Okay, it's called a receptor. So this receptor is a perfect match for mannose. And so what happens is when you have yeast cells that have mannose in their cell wall, because all yeast cells do, and they have this receptor on their cell surface also, the receptors on one cell bind to the mannose on the neighboring cell. That cell's receptor binds to the mannose on this cell, and that's what makes these stick together. Okay? This receptor protein is called FLO1. And so if you have cells that have a normal FLO1 gene, those cells are sticky to one another because they're making this protein that binds the mannose and that makes these clumps of cells. If you have a mutant FLO1 gene, then no receptor is made. The mannose is still there, obviously. Okay? But these cells don't stick to each other anymore because they don't have these receptors there to bind the mannose. Okay? And so the yeast strains that are used to make clear beers are actually mutants. They don't have a functioning FLO1 gene. And the yeast strains that are, sorry, cloudy beers. The yeast strains that are used for the clear beers are sort of wild type yeast. They're normal yeast, and they clump up and sink to the bottom. Okay? Now what's interesting about the FLO1 gene is that it has a repeated sequence in it. Okay, so if I draw a schematic of the FLO1 gene, or part of the FLO1 gene, I have the start codon over here, so it would start ATG and so on and so on. Stop codon over here, okay? And in the middle of this coding sequence is the same sequence repeated 18 times, okay? And as you know, DNA polymerase has problems with repeated sequences, okay? So when it copies this gene, every so often, this number 18 is going to increase or decrease, okay? So now there are a bunch of yeast strains that exist that have different numbers of repeats right here. Sort of the canonical number is 18, but any given yeast strain could have fewer repeats or more repeats in the gene. And so what was done a few years ago was to look at a series of these strains, okay, from really cloudy ones to really clear ones. And just by shining a light through these test tubes and seeing how much light gets through the other side, you can quantify how cloudy these things are, okay? And that's what's shown on this axis, amount of light passing through, okay? So that's a measure of clumpiness, because if all of the light passes through, then the yeast had to be clumpy. They sunk to the bottom. And if less of the light passes through, it means it was cloudy. The yeast were not clumping with each other, okay? And so that's what's on this axis of the graph. And what's on this axis of the graph is the number of repeats in their FLO1 gene, okay? And so what you see, these are strains A to H, from cloudy to clear, clumpy to not clumpy, is there's a really nice correlation between how many repeats there are in the gene and how much light passes through, okay? So you have strain A, very little light passes through, and it has the smallest number of repeats in the gene. Strain H, lots of light passes through, and it has the most repeats in the gene, okay? So what we can infer from this is that the more repeats you have, the stickier the cells are to one another, okay? So you get the really clumpy strain with the highest number of repeats, and the really unclumpy strain with the lowest number of repeats, and it's actually a quantitative relationship, okay? The more repeats you have, the more you tend to be clumpy, okay? So that's sort of cool in and of itself, but it relates to something that really has important implications for humans, okay? And the reason it has important implications for humans is there are a number of genes in humans that have the same basic structure, where they have repeats in the middle, okay? And probably the one that's best known to you is the cause of Huntington's disease, okay? So the Huntington's disease gene has a CAG repeat in the middle of it, okay? And that can be repeated anywhere from 6 to 121 times, depending on the individual, okay? If an individual has between 6 and 35 repeats, they're normal. They don't have Huntington's disease. But if they have more than 36 repeats, they're at risk for the disease. And the higher the number of repeats, the more severe the disease tends to be, okay? So in a way, this is the human equivalent of this very simple example from yeast. That is, a small number of repeats is associated with one phenotype. In humans, it's normal function. And a high number of repeats is associated with disease, okay? And these types of repeats are really hard for DNA polymerase to copy, right, as I said. And you can see that by just looking at parents and their offspring, okay? So as you might expect, the way we can tell how many repeats an individual has is by running a gel, okay? Because that tells us the length of a particular DNA sequence. And if your DNA sequence is the sequence surrounding these repeats, the longer it is, the more repeats you have. The shorter it is, the fewer repeats you have, okay? So what we can do is take the DNA from a particular individual and see how many repeats their Huntington's gene has. So what's shown on this gel, and you should be sort of starting to get used to what these gels look like and how frustrating they are to produce, right, is a size marker, okay? That's just sort of a standard that tells us, how many repeats we're actually looking at, and you can see the numbers over here, and that's the borderline, okay? If you have fewer than 35, you're okay, and if you have more than 35, you're not okay. You're at risk for the disease, okay? And so this is a male, a female, a father and a daughter, right, in sort of pedigree notation. And if they're colored in, that means they have the disease. And if they're not colored in, that means they don't have the disease. So if we look at this male that doesn't have the disease, we see these two bands. One is at about 18, and the other one's at about 12 or so. Why are there two bands? Two different. So this gel is not really cutting, okay? So the DNA sequence you're looking at is sort of, let's imagine it's the full sequence of the gene.
Oh, the part
That's
that's not the repeats?
not it either. Okay, these are good guesses. Keep guessing. Why would each individual have two bands here? It's sort of a simpler answer. One from the mother and one from the father. Okay, we each have two copies of every gene. Okay, so one of these came from the person's mother and one of these came from the person's father, and you can see that let's say the father passed on the gene with 18 repeats and the mother passed on the gene with 12 repeats, or the other way around. Likewise, for this male individual who doesn't have the disease, one parent passed on the copy of the gene that has borderline, so somewhere around 34 copies of the gene, copies of the repeat, and the other parent had sort of a more normal copy with like 14 versions of the repeat. Okay, and so this guy's borderline, right? And so you can imagine that if this guy had children and this guy had children, which one's children would be more at risk for getting the disease? This one, because in one generation, this can actually change to a higher number of repeats or a lower number of repeats. But if it changes to a higher number of repeats, then that child will have the disease. Okay? So here's an individual that has the disease. And you can see the two bands are here, which is a normal copy of the gene, and here, where it's somewhere around 38 repeats or so. So 38 repeats is enough to cause the disease. It'll be sort of a more mild form of the disease, but it's still a very, very serious disease. You can see how just sort of a small difference in the DNA, a few bases strung together, can be the difference between healthy and diseased. Right? If we look at this father, you can see that one copy of the gene is normal and one copy of the gene has a large number of repeats, somewhere around 50 repeats. Okay? So before we look at the child of this father, just by looking at this individual and this individual, you can tell me if this disease version of the gene is recessive or dominant. So recessive or dominant? That is, do you need one bad copy of the gene or two bad copies of the gene to have the disease? You just need one bad copy of the gene to have the disease, right? This individual just has that bad copy, and this individual just has that bad copy, okay? And if you just need one bad copy to have the disease, then that copy is dominant, okay? Now look at this father having a daughter, okay? If this were a normal gene, you would expect to see that the daughter would inherit either this band or this band, right? But instead, she has two different bands, two different size bands. She has one that's about 86 copies and one that's about 16 copies, okay? Which one of these did she get from her father? You can't tell for sure, but which is more likely? This one, okay? This one probably from the mother, okay? And look at what happened. In the course of one generation, that's just one meiosis, one forming of gametes, you go from 55 to 86 copies. Okay, that means the polymerase was going along, and instead of faithfully copying 55 copies, it went along, and then it looped back, and then it copied 31 of those repeats again. Okay, and that's how you expand up from 55 to 86 just in the course of one generation. Okay? Now this individual is going to have a much more severe form of the disease than this individual, who's going to have a much more severe form of the disease than this individual. Yeah.
So the fact that they have the disease is because of the repeats, but because the difficulty the DNA polymerase has in copying.
So we'll get to the actual cause of the disease on the next slide, right? But so the question is, well, what causes the disease, right? We know that this gene can change in copy number, but if this gene was not important in the life of the cell or the organism, then these changes would happen and it wouldn't matter because it wouldn't cause any disease. But the point is that this repeat is in the middle of a gene, in the middle of the coding sequence of a gene, and so by expanding up and down, it might actually change the function of the protein that gene makes. The same way the FLO1 protein from yeast, its function changes depending on how many repeats it has. The longer the protein, the more sticky it is. The shorter the protein, the less sticky it is. Okay? And it turns out it's exactly the same kind of thing that's going on with this protein. The longer it is, the more likely it is to stick to other proteins. Okay, so this is a sort of rendering of what that would look like. These HTT fragments are basically proteins from this gene. Okay? The disease is called Huntington disease. The protein is called huntingtin. Okay, so these are a bunch of fragments of huntingtin protein. And the longer they are, the more repeats they have, the more likely they are to sort of form these like tangled messes. Okay? And in general, you don't want your proteins to be in a tangled mess. Okay? Proteins have jobs to do. Okay? And so if your proteins are in a tangled mess, it's unlikely they're going to be performing their job properly. Okay? So these tangled messes we usually call aggregates. Okay? But you can see it's a tangled, rigid grouping of altered protein fragments. Okay? And so what's thought to actually cause disease is that this protein is made in nerve cells, in neurons. Okay? And so here's a picture of a neuron. It doesn't look like a normal cell. A normal cell would have this sort of cell body and nucleus, okay? But neurons, as you know, are forming connections with each other. They have these long axons and dendrites that make those connections. So you can see they have these axons. So this would connect to some other neuron or maybe a muscle. And all of these projections are what gives you all these connections in your brain that allows you to do important things like think. In these neurons, these axons and dendrites are actually fairly narrow, okay, as depicted here. And what can happen is that these tangled masses of huntingtin protein can actually get stuck, okay? And when they get stuck, it's called an inclusion body, right? And that can disrupt the function of these cells, right? Later on, these tangled bits of protein can actually get inside the nucleus and cause problems there, okay? And so the basic idea is that when you have these tangled huntingtin proteins in your nerve cells stop working properly, okay? And ultimately will die, okay? And when your nerve cells in your brain stop working properly and start dying, then you have very, very serious disease, okay? You lose motor control, you lose cognitive capacity, and eventually you die. There are a set of diseases like this that are like Huntington's disease. There's a repeat of 3 nucleotides somewhere in the gene encoding for a protein that's usually expressed somewhere in the nervous system. Okay, so Huntington's disease is a CAG repeat. The normal range is 6 to 35 CAGs, and at risk for the disease is somewhere above 36 repeats. Okay. All of these other diseases are so-called trinucleotide repeat diseases because, again, they have 3 nucleotides that are repeated. Why do you think it's 3? It's a codon, right? If you had a 4 nucleotide repeat, the protein sequence would basically be out of frame. And so if you added an extra repeat, you would not only get a longer protein, but you'd get the wrong sequence in the protein. Whereas if a CAG repeats, so CAG codes for glutamine, right? So if CAG just repeats, you will either have 20 copies of 20 glutamine amino acids in a row, or you'll have 100 glutamine amino acids in a row, but the rest of the protein will still be the same.
Can I ask why the spinocerebellar
Yeah, so I mean, there's no necessary reason why there would be a really firm breakpoint between disease and not disease. Okay? What this means is that some individuals with 44 repeats never develop the disease, but some individuals with 39 repeats do. Okay? What you can see is some of these repeats expand to amazing lengths. Okay, so if you look at myotonic dystrophy, it's a CTG repeat. Normal range is 5 to 37. Some individuals have 3,000 repeats. Okay, their gene just reads CTG, 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 CTG 3,000 times. Okay. All of these tend to be neurological diseases with sort of similar overlapping symptoms with Huntington's disease because they basically result in damage to your nervous system.
So if the DNA polymerase could correctly copy the repeats, then it wouldn't become an issue because they wouldn't talk about them
That,
despite the repeats.
well, so that's exactly right. So if a parent had 20 repeats of the Huntington's gene, right, and the DNA polymerase was perfectly good at copying those repeats, then the disease would never develop because you would never get any individuals with the larger number of repeats. And so in a sense, the most proximal cause of the disease is these proteins getting stuck in neurons, right? The less proximal cause is that the DNA polymerase doesn't work properly and causes genes with more repeats than they should. But they're separated in a sense. Just not being able to copy your DNA properly is not a cause of disease, right? It's just a cause of a gene changing to the point where it causes disease. Okay? And one of the sort of surprising, amazing things about our genomes is that we have lots of these kind of repeats. You know, you think it'd be, if there was someone designing organisms and you knew that DNA polymerase had trouble getting through CAG, 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 you wouldn't put CAG, CAG, CAG in an important gene. Okay, it would be a stupid design principle to make your most important genes have this kind of fragility where they would get copied wrong all the time. Now, it turns out that a lot of our important genes actually do have repeats. And one class of genes that tends to have repeats like this are transcription factors. So you've seen examples of transcription factors, although I might not have called them transcription factors. Transcription factors are proteins that bind to DNA at particular sequences and that control whether the gene that's there gets transcribed or not. Okay? And so one example of a transcription factor that you've seen is the lac repressor, which binds to the operator of the lac operon and prevents transcription from happening. Okay? So the lac repressor is an example of a transcription factor, and it's more specifically an example of a repressor transcription factor. There are also transcription factors that are activators. They also bind to a sequence near the beginning of a gene, and their presence there makes it more likely that RNA polymerase will come and start transcribing the gene. These are called activators, okay? And there's an activator for the lac operon that is controlled by the presence of glucose, okay? It turns out that activators tend to have repeated amino acids. In fact, one of the favorite sequences within activators is glutamine, 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 okay? And so this is a problem, right? If activators have these glutamines in them and these are prone to mutation, then it could be that our activator proteins are changing all the time as parents have progeny. And in some cases, this can cause problems. And as you'll see, in dogs, it can actually cause variation that we see. Okay, it doesn't always cause disease, right? And the reason these proteins are important activators are that they control the activity of other genes, right? And in general, proteins that control the activity of other genes are used during developmental processes. Okay, you can imagine that in going from an embryo to an adult, there are a lot of gene changes that have to happen, and there have to be proteins that orchestrate these changes, right? Embryos need different genes as they're developing from adults' genes. And so these transcription factors control these developmental processes, and you can imagine that if a transcription factor doesn't do its job properly, you can have a large effect on an observable trait, and you'll see some examples right now. Okay? So, back to dogs. Here are the observations about dogs. First, the reason we're studying them is that they differ dramatically in observable traits. Okay? We know these differences arose very recently in the last few thousand years. Okay? We know repeat lengths change very rapidly, so maybe this is the type of mutation that's relevant to these differences. We know that these repeats tend to be found in genes important for development, so that again lends credence to the idea that maybe a simple mutation in one of these genes can actually cause a big difference in how the animals look. Okay? And that's because when you have a developmental change, it can really dramatically alter a trait. So a few years ago, these guys came up with the hypothesis that repeat length changes in developmentally important genes, in particular transcriptional activators, might be responsible for the evolution of all these different differences between dogs. Okay? And so they figured out an experiment to test this. And in essence, the experiment is very simple. Okay? Basically, what they did was they took dogs from different dog breeds, and they got DNA sequences from them, but the DNA sequences they got were from transcriptional activator genes, and they were from transcriptional activator genes that do have these trinucleotide repeats. And so they got the sequence that sort of spanned those trinucleotide repeats so that they could tell how long those repeats were. Okay? Similar to how you would want to tell in a Huntington family, a family where Huntington's disease is segregating, whether they might be at risk. The same as when you're doing that yeast experiment, you can tell how many repeats there are, and that'll tell you how cloudy your beer is going to be. They get sequences like that, and they look for correlations between the number of repeats and a particular trait in a particular dog. Okay? And so the first example I want to show you is the ALX4 gene, okay, which it turns out has a different repeat length in one breed, the Great Pyrenees. Okay? So this is a gel, and each lane is a different dog breed, okay, and they're all the same, all the same, all the same, and then there's one dog breed that has a lower band, so that means it's shorter piece of DNA, right? Fewer repeats, okay? And so they can actually get the DNA sequence from that breed and see that it's actually a change in this microsatellite, okay? So this is the sequence of any one of the other dog breeds. You can sort of see the DNA sequence chromatogram, so that would be CC, CC, GC, A, GC, A, GC, GC, A, and so on, so on, so on, so on. Okay? So there's this repeat here that's either CCG, CAG, and so on, repeated a few times. Okay? And what happened in this one dog breed is that from here to here got deleted. Okay? That is the polymerase had a problem here, and it skipped right from here to here. Okay? And you can see that this is the sequence from the Great Pyrenees, because the sequence after, this is the single letter amino acid code, so these are codons, right? So after this deletion is AQPP, alanine, glutamine, proline, proline, okay? And instead of being here, that sequence is way over here, alanine, glutamine, proline, proline. Okay, so you have this shifting of the sequence where this part of it got chopped out, and only in one breed. So then what you could do is you say, well, what's special about that breed, just by looking at it, okay? And it turns out they have a very, very strange feature, okay? So you can go to the American Kennel Club website and look up everything there is to know about the Great Pyrenees breed and what it takes to be a Great Pyrenees that would win the Westminster Dog Show, okay? And one of the things that can get you disqualified, don't even get in the door with your Great Pyrenees, is what's called an absence of a double dew claw on each rear leg. Okay? So that's a fault. So if this dog does not have a double dew claw, it's out. It's not a Great Pyrenees as far as the American Kennel Club is concerned. So what is a double dew claw? Okay? So the dew claw is one of the toes on a dog, right? And the Great Pyrenees has two here, okay? Unlike other breeds, okay? Normal dogs have 5 toes, okay? Great Pyrenees has 6 toes. 1, 2, 3, 4, 5, 6. Okay, it has this double dew claw, right? You can make a mutation in the same gene that is unique in repeat size to the Great Pyrenees in a mouse, okay? And the phenotype you get by deleting this gene in a mouse is a duplication of the toe. Okay, so this is now a picture of a mouse skeleton from a mouse that has a double dew claw. Okay, So that's really, really dramatic evidence that the reason the Great Pyrenees breed has a double dew claw is that it has an alteration in this gene, okay? And the alteration is a very simple one. The alteration is that it has a little bit of sequence missing from this gene, okay? It has a shorter repeat length. Okay? Here's another example. So this is a skull of a human patient with what's called cleidocranial dysplasia, or CCD. Okay? And one of the sort of diagnostic features of this disease is skeletal malformations of various kinds, but a very, very, very bulging forehead. Okay? Very, very, very serious disease, right? It's now known that the disease is caused by a mutation in this gene called RUNX2. Okay? So one of the things these guys did with dog breeds is they looked for cases where the skull morphology is interesting. Okay? And one of the most interesting cases of skull morphology change is in the bull terrier. Okay? So you've probably seen bull terriers. They have this really sort of strong snout, and it has this interesting shape that sort of bends down. Okay? And what's interesting about the bull terrier is that it wasn't always like that. Okay? And in fact, the standard changed over the last few decades, where it used to be a bull terrier should have a straight snout, but over the years it became more and more desirable to have a bent snout. Okay? And so if you look at skulls of bull terriers from 1931, 1950, and 1976, you can see how dramatic artificial selection can be. You know, just by breeders choosing bull terriers with more downward-facing dogs, over the course of, 45 years, you go from a skull that looks like this to a skull that looks like that. Okay? All the same breed, just at different points in time. Okay? And so the question that these guys ask is, well, maybe a change like this could be caused by a mutation in a gene like RUNX2, which is known to affect the shape of the skull. Okay? So what they did was they quantify, and RUNX2 has repeats in it. Okay? So what they did was for a bunch of breeds, they quantified how many repeats RUNX2 has, and they quantified the shape of the snout. Okay? And so the way you nowadays quantify the shape of the snout is you take these really high-resolution 3D reconstructions of skulls. Okay? So you have these skulls, and you have this special laser setup that can view it in all directions and scan the whole skull, and basically give you something with much, much, much higher resolution than any X-ray. Okay? So you basically get the entire 3D reconstruction of what the skull looks like. And you can see this is what a wolf skull looks like, and this is what one particular dog skull looks like, obviously not to scale. This is a Chihuahua. Okay? So a Chihuahua is an example where the skull, instead of bending down, bends up. Okay? So what these guys did was they gathered a bunch of different skulls and DNA from these different breeds, from everything from a Chihuahua, where the skull bends up, to a bull terrier, where the skull bends down. And you could just measure this amount of bending. So this would be sort of a positive curvature and negative curvature, or the other way around. It doesn't really matter. But you can have an axis here for how curved the snout is. And then you can just count the number of CAG repeats in the RUNX2 gene. And just like in the example with FLO1, where the number of repeats correlates with this phenotype, here you have also a very strong correlation. So the more repeats of this RUNX2 gene you have, the more likely you're going to have a bull terrier type snout that bends down. The fewer repeats of this gene you have, the more likely you have a Chihuahua-like snout that bends up. Okay? So this, again, is really amazing confirmation or support for the idea that the differences between dog breeds occurred in genes that are more prone to mutate rapidly, and that these genes that encode transcriptional activators that tend to have these repeats in them are a real likely target for these, where in the DNA these changes happen. I want to go through just a couple more examples of very recent studies. Okay? A lot of these studies on dogs associating traits with particular DNA sequences are only a few years old. Okay? So here are two examples from 2007. where particular genes were found to be causative of the differences between dog breeds. Okay, so here you have a big dog and a small dog. Okay, I don't know if you can, in the back, if you actually saw that there was a dog here, right? So the point is that dogs are amazingly variable in size. They're as far as species go, they're mammalian species, they're the most variable in terms of the size of members of the same species. Okay? It turns out that the difference in size between a dog like this and a dog like this is mostly due to one single nucleotide in their genome changing. Okay? And that single nucleotide is in a gene called IGF-1. That stands for insulin-like growth factor 1. Okay? So it's a gene that encodes a protein that's a growth factor. Okay? So as you might imagine, the small dogs don't make as much of that growth factor as the big dogs do. Or the growth factor that they do make is less effective than the big dogs. Okay? And so it turns out that if you look at all of the small dogs and compare them to all of the big dogs, it's the same mutation. That suggests that all of the small dogs are relatives of each other, that they were sort of, there was an original small dog and then the other small dog breeds were bred from those. Here's another example of just a change in 2 bases in one gene that causes a really, really dramatic phenotype. So this is a normal whippet dog, okay? And this is what's called a bully whippet. For a reason you can probably understand, this dog is like this hulking muscular mass of a dog, okay? The reason it's a hulking muscular mass of a dog is that 2 of the bases in one of its genes are missing. The gene is called myostatin. Myostatin is a negative regulator of muscle production, okay? And so if you delete this gene or make it non-functional by deleting 2 of its base pairs, then that myostatin gene, which normally puts the brakes on muscle development, is no longer putting the brakes on muscle development, and you get this like seriously over-muscled dog. Okay? You can imagine that, you know, sort of nefarious Olympic committees in various countries might be figuring out a way to do this to people. This is a cool example, which is from last year, of figuring out why the coats of dogs look different. So you can have like an Irish Water Spaniel which has this really curly hair, or a Bichon Frise which has really, really curly hair. Some dogs have these things called furnishings, right? The sort of extra like mustache type things on them. Some have really wiry coats, some have less wiry coats, some have really long hair, some have really short hair. And this combination of 3 genes basically determines this sort of range of differences. So in a dog like a Basset Hound, which just has, you know, really short hair, it has normal versions of these 3 genes. Okay? In an Australian Terrier, which has short hair but it's wiry, it has a mutation in this one gene here, arspondin 2. If you have an Airedale Terrier, which is wiry and curly, then it has a mutation in 2 of the genes. If you have a Golden Retriever, which is sort of the opposite of that, it's long hair and it's not curly, then it has a mutation in this gene. If you have a Bearded Collie, which has long, straight hair, but it has also these furnishings, okay, it has a mutation in these 2 genes. Irish Water Spaniel, these 2 genes. And the Bichon Frise is a mutant for all 3 genes. It has long, curly hair plus these furnishings, okay? And so basically, if you gave me the genotype at these 3 genes, I could tell you whether your dog is more likely to look like this, or look like this, or look like this. And likewise, if you gave me a dog that had sort of furnishings and wiry hair, I could tell you what its genotype is likely to be. What's interesting is that, you know, these mutations are in genes that probably play a big role in human development too. Okay? So FGF5 is in the gene for fibroblast growth factor. So another growth factor. This growth factor is associated with long hair, growing hair. Arspondin 2 is also known from experimental studies to be involved in hair follicle development. Okay? And KRT71 is a keratin gene. As you know, keratin is a major component of your hair. Okay? So this all sort of starts to make sense, and it's amazing how these DNA studies can really just sort of explain why this dog looks like that and that dog looks like that. And if you add in other things like skull morphology, color of the hair, you can start putting together how a relatively reasonably small number of genes can actually explain a difference in appearance between these two things which look so different from one another. And just one final example is, again, a really big change from a single mutation, okay? And this is a mutation in the FOXI3 gene. This is a transcription factor gene. And just this single mutation, which is dominant, gives you that going to that, okay? So these two dogs, for all intents and purposes, have identical genomes except for one nucleotide. And that nucleotide makes these guys hairless and these guys not hairless.